This is the SARS-CoV-2 mutation tracking and genomic data visualization talk. And the speaker is Albert Chen, an associate computational biologist in the vector engineering group at the Broad Institute of MIT and Harvard. Albert's the project lead on the COVID-19 project that he's going to talk about and contributes to gene therapy efforts of the vector engineering group. He has prior experience in web development. In the last 5 years is splitting his time between dry lab, which is in essence computer work, and wet lab, which is actually, real lab work. So Albert, I'm gonna hand off the screen to you.
Great, thanks, Mark. Can everyone see the presentation okay?
Mm-hmm.
Awesome. Should we wait till the 7:30 or just start now?
I think you can start now.
Okay, great. Yeah, so thanks Mark for the introduction. By the way, I'll be open to questions anytime during the talk. You can just unmute, or there's like a raise your hand feature. I haven't used it, but we might go over some biology concepts that might not be familiar to some of you, so please feel free to ask questions. I think I tried to keep the talk at 30 minutes, but I think I'm going to go over a bit. That's going to be okay, right, Mark? Okay, so just as a disclosure, our COVIDCG team has a sponsored research agreement with AstraZeneca, who is a COVID-19 vaccine and therapeutics developer. And Xinghei Zhang, who is part of the COVIDCG team, is also a co-founder of Fusion Genomics Corp, which is does develop some molecular diagnostics for COVID-19 testing. Okay, so I'll be splitting today's talk up into 3 parts. I'll be actually spending most of my time talking about sort of COVID-19, SARS-CoV-2 biology, and these rising variants that you might have heard about on the news. Then I'll go through an abbreviated story about how we built COVIDCG. So this is more of like a technical perspective on things, and then I'll end with some perspective on how I think the data science community and this great community can play a part in advancing science, biology, therapeutics, and global health. Play a big role in biology and data moving forward. Okay, so yeah, starting out with some introduction on COVID-19. So COVID is not going away. There's a wide range of predictions last year, but I think it's clear now that we won't be out of the woods for a while. I pulled this data from about 2 weeks ago, so it's a little bit out of date, but even with 70 million Americans fully vaccinated and 120 million receiving their first dose, we're still at a rate of about 50,000 new cases per day. And I think there's been some news going around how we might not reach herd immunity depending on vaccine hesitancy and other factors. Can't really speak to making any predictions, but I think it's clear that it's not just going to go away in the next year. So this is going to stay with us. And COVID is also not just a problem in the US, it's a global problem. Viruses don't really care about national borders, and while COVID is getting better in the US relative to our big peak in January, it is getting worse around the world. So in particular, we have Brazil and India are going through their worst waves in the pandemic. Just like our worst wave was back in December, January, February, they're going through their worst waves right now. But yeah, why does this matter? Why do we care about other countries? We live in the US, or most of us. This is a PyData Boston group. It's because SARS-CoV-2, this word up here, this is the name of the virus that causes COVID-19, so I'll be saying that term a lot. SARS-CoV-2 is mutating, and the more human hosts it infects, the more chances it has to mutate. Already we have strains imported into the US, which were initially detected abroad. One of the most prominent ones is this UK variant. The general scientific consensus is that this variant is more transmissible between people, and it's also now the most common source of new infections in the US, and this was originally detected in the UK. And also, in addition to being more transmissible, this strain has also been reported to be deadlier than the original SARS-CoV-2. We have this Brazilian strain. So this strain has become the dominant variant in Brazil's epidemic. And in this article here, the authors hypothesized that this variant could potentially be reinfecting patients that have previously contracted SARS-CoV-2. It has generally higher transmissibility and more antibody evasion. You also may have seen the South African variant on the news. And in this current paper, the authors show that the AstraZeneca vaccine, which is not approved in the US, but is approved elsewhere, actually just is not very effective at dealing with the South African variant. And when I made these slides initially, I just had these 3 samples, but even just within the past 2 weeks, India's recent COVID-19 surge has brought to light this new Indian variant, which we don't know a whole lot about, but we're definitely keeping our eyes on. So again, these mutants are not, these variants are not just around the world. They've already made it into the US, all 4 of the ones that I've showed. This B.1.1.7, which is also known as this UK variant, you'll see here in North America, that is now the most predominant source of new infections. And we have other variants like the Brazil variant that are rising. I did not show the South African and Indian variant, but they do exist in the US, just in very small numbers as of right now. And I don't, just highlighting that in North America. I don't really want to be too doom and gloom about things, about how the world's ending. I think one silver lining from this pandemic is that the development of these mRNA vaccines, the Pfizer-BioNTech vaccine and the Moderna vaccine, I think it's amazing. These mRNA vaccines work really, really, really well. And I mentioned that the South African variant renders the AstraZeneca vaccine, which is not an mRNA vaccine, ineffective. This Moderna did an internal study in the lab, so not on humans, which showed that the neutralizing activity of their vaccine went down about 6 times when tested against the South African variant. Again, this is in a lab setting, so not totally translatable to the real world. But I think they found that in this lab setting, even though their vaccine worked 6 times less well, it was still within the effective range, which means that, hypothetically, that the Moderna mRNA vaccine would still provide full protection to people exposed to the South African variant, which is great. It works so well that it can take a 6-fold hit and still work. And we see a similar result with the Pfizer-BioNTech vaccine when tested against the UK variant as well. And another amazing feature of these mRNA vaccines is that they can be developed and adapted really quickly. So a little known fact is that the Moderna mRNA vaccine was designed in just 2 days. The rest of the time was spent in production and in clinical trials. And since modifying these mRNA vaccines is just modifying some of this genetic mRNA code, we can adapt these existing vaccines to fight these new variants. So Moderna right now is piloting one version of its vaccine to protect against the South African variant, which I just mentioned before. Okay, so I guess the point of me talking about these vaccines is that in these variants is that this SARS-CoV-2, the virus that causes COVID-19, is evolving, which means that everything we do to fight COVID-19 must also evolve. And our pandemic response is not just limited to the vaccines I was just talking about. COVID-19 tests, so getting tested for COVID-19, relies on knowing a certain portion of the virus's genetic code. So if this genetic code mutates, then so must our diagnostic test. And while everyone's really focused on vaccines at the moment, I think our diagnostic tests are really critical for being able to get our lives all back to normal again. We're all developing therapeutics, which differ from vaccines in that these therapeutic treatments are designed to blunt severe COVID-19 symptoms. So these are for people that have already experiencing severe COVID-19 symptoms that have already gotten it, not for preventing in the first place. I think one really prominent example of a therapeutic was this Regeneron antibody cocktail that former President Trump received when he contracted the virus. And also on public policy front. So I think staying on top of the virus can really help inform public policy. If we're aware of a concerning variant abroad, then we can implement stricter travel controls or better quarantine policies to prevent its spread to the US. We have not been particularly successful so far, but I think this is something that we could make an effort to in the future if we are aware of these variants earlier. Okay, so yeah, before we go on, I just want to get this definition out of the way about what's a mutation and what's a variant. So the SARS-CoV-2 genome is 30,000 bases long, 30,000 characters long, where a character is A, T, C, or G, And it's kind of funny to me because biologists express this genetic information in, I think, similar terms as to computer scientists explain their data. So at 30,000 bases, we usually shorten this to say it's 30 KB, where KB stands for kilobases, almost like kilobyte, except it's not a byte, it's a letter in this case. And yeah, in terms of evolution, viruses are just like every other living organism on this planet. They mutate and evolve and are affected by natural selection. You know, most of these times, so I'm just showing here some mutations on this genome. Most of these times, these mutations do nothing. The mutations are never intentional, they're always random, but sometimes they will randomly change the behavior of the virus, and these can be selected for via natural selection. So, and again, just like us, these mutations are hereditary and they can accumulate as the pandemic continues. So a variant or strain, as you might hear called, is a set of viruses that are evolutionarily related. So viruses with a common ancestor. So here I'm showing a plot from this next strain site where we have sampled a bunch of these different viruses all now, and you can see that they all share this common ancestor here in the middle, and they're all branching out into these diverse groups of variants and strains. And I also just want to address a common misconception about sequencing versus COVID-19 testing. So a common misconception is that when you take a COVID test, that this viral genome is actually sequenced. COVID testing is actually much simpler than genome sequencing, as we only rely on detecting about 100 base pair segment of the SARS-CoV-2 genome. This is about only like 0.3% of the genome. And these tests will only give a binary result. It's either positive or negative. So as a result, COVID tests are really cheap to run and they can be massively scaled up. Our facility at the Broad Institute has about like a one-day record of 150,000 COVID cases, or COVID tests, sorry. And yeah, sometimes these positive COVID tests are sent to sequencing centers. These are physically different locations than these testing centers. And the cost is usually higher, the throughput's usually lower, and the turnaround time is usually longer. So as a result, the majority of positive COVID-19 cases are not sequenced. In the US, that percentage is about 0.7%. So we only sequence about 7 out of every 1,000 COVID cases. And this sequencing actually happens at various different efforts around the world. So here I'm showing a map where the darker colors represent more sequencing done relative to the number of positive COVID-19 cases. So whereas the US sequences about 7 out of 1,000 positive COVID-19 cases, the UK sequence is almost 10 times the amount. And we have countries that are doing even more sequencing, where Australia is sequencing almost half of their positive COVID cases. So this is really advantageous for them being able to stay on top of what's happening genetically with COVID-19. And we also have some countries like those I highlighted in the beginning, Brazil and India, who are sequencing much less relative to the magnitude of their pandemics. And again, this makes it a bit harder to know what exactly is going on. Yeah, and our reporting of this data has actually attracted some press attention. And if you're interested in learning more, we display this data and in a lot more detail on the front page of our COVIDCG website. Okay, so that's the introduction. I think now you all, this talk is about this tool that we built, COVIDCG. So what does our site actually do? I'm just showing here a screenshot of, I think, one of the most powerful tools in our site is just a way to, filter data. Oh, sorry. I think I just noticed that there are some questions in the chat. I'll address those right after these few slides. Sorry. Oops, my bad. So yeah, I think there's the existing interface for selecting these genomes we wanted to just improve on this. So we hope we have this more intuitive interface for really drilling down into the data to have really detailed answers. For example, if you're a public health policy professional in Massachusetts, you want to know what variants of COVID are swimming around Massachusetts in the last month, and you can answer these questions in a really targeted way. So yeah, once the data is selected, we here present an overview of these selected genomes. Here I'm selecting this B.1.1.7 variant, which is the UK variant. Sorry, let me just replay that video. You can also see that when we have these data, it starts out as counts, and these counts can be sort of biased depending on how much sequencing is done on the given day. So we have the option to change these to percentages. And then once we've also selected this data, we can also compare between locations. So here I've selected a bunch of various states and just showing that we have various levels of this UK variant in different states. So for example, I think it's the highest in Florida and the lowest in California as of about 2 weeks ago. And I think some of the most powerful features of our COVIDCG site is actually the data downloads, not necessarily visualization parts, but we've with this we've noticed that researchers and scientists have been the most excited just about getting their hands on the data and being able to run their own plots, make their own plots and run their own models. Okay, sorry for skipping these questions. I'll get to them. So Ben asked, how do they decide which variants to test the vaccines against? Are they testing everything or just ones with a certain number of cases? That's a good question. I'm not sure what the selection process is. I think whenever one of these, at least internally in these companies, whenever one of these variants pops up worldwide, they will immediately test their vaccine against it or their therapeutics against it to make sure it still works. And I think if maybe it starts to show signs of not working, just like that Moderna study did, then they will maybe target it for what we might in the future call like a booster shot. But again, I'm not 100% sure. Adam asked, why are we not sequencing more? That's a great question. So going back to this previous slide, you'll see that there's a really wide range of sequencing efforts between countries. I think a lot of this is actually due to sort of incentives in science and government policy. So the UK, and I'm not 100% sure about these other countries, but I know for the UK that the government has actually invested a lot into sequencing. There is actually this big debate about this UK variant, which I'm talking about, which it was initially detected in the UK because the UK sequences so much of their virus. We don't actually know if it originated in the UK. It may very well have originated in a different country, jumped to the UK, and the UK caught it because they're sequencing so much. And this is just one of the benefits of staying on top of things and sequencing a lot. So yeah, to answer your question, I think a lot of it has to do with just incentives. I think sequencing is expensive. Scientists will not spend their money and their free time sequencing COVID-19 unless they can get something out of it. And this is something we've also seen with a lot of groups around the world that will sequence a lot of genomes, but wait a couple of months before they release it because they want to get a paper out. And yeah, I think that the UK has done a really good job of aligning the incentives with the public and trying to get this data out as quickly as possible. So yeah, I think certainly the US can do a lot better in terms of sequencing more. Yeah, so Christine asked, what's the purpose of sequencing to learn exactly which variant it is? Yeah, that's exactly right. I think going back to this slide, it's just the virus is going to change. I think right now, the virus is still relatively similar to the first version of the virus that came out of Wuhan about a year ago. I think in next year or in 2 years or 3 years, if COVID-19 is still sticking around, that it will change dramatically in that all the things that we rely on to fight COVID-19 right now, like our COVID tests that you might take at local testing centers or therapeutics or even our vaccines might need to be updated. So we just need to stay on top of these variants and know what they are so that we can really quickly respond to these mutations. Yeah, sorry. Also, feel free to unmute and ask me the questions. I don't mind being interrupted at all.
So is there a plugin where you can also look for the gene? That the mutation is in S or M or N or E, which gene?
Yeah, so all the let me go back to the site. Sorry for the blurry image. Yeah, all the examples I'm showing here are in the spike gene because that's the most therapeutically relevant gene. But in our site, we allow selecting for any of the genes in the SARS-CoV-2 genomes. Yes, so the E to S, any of the proteins in the ORFs. We also have additional, if we go back to the selector, you'll see that we allow selecting a gene. You can select by protein. We allow searching by primers and probes. So for people that are developing diagnostics, they can make sure that their diagnostics are still working within their local area that they're testing their populations in. And we also allow for matching like custom coordinates or sequences. So we allow selecting any arbitrary span of the coronavirus genome. Yeah.
Right. And can you submit your sequence? Like if you have a SARS-CoV sequence, can you upload it and compare it with your data set?
So we pull from the GISAID database, which is this global database where everyone submits their sequences. So we would only get your data if you would submit to this GISAID database first.
Okay.
We also have a version of the site that pulls off of GenBank, which is another database of SARS-CoV-2 sequences, but it's a lot smaller. Sorry, Christine, I see your question. I'll get to you in a second. But yeah, I think an important thing to note is that this COVIDCG website is also open source. So if you want to run this application locally, that you can do this on your computer and load your own sequences if you want. All right, Christine, I saw you had a question.
Hi, I have a follow-up question to the one that I just asked.
Yeah, sure.
So the reason to do this, sorry, I'm not familiar with biology, but the reason to do the sequencing is to know exactly which variant it is. So yes. I was wondering, so earlier you had a few slides about every part of the world, like, I don't know, maybe in the past 12 months, for example, in the US or in North America, the percentage of cases that is actually like B7, the UK variant. Yeah.
Yeah.
So basically this slide, the data here, right, is not like 100% accurate because you said we are not really doing the sequencing.
Yeah, you're right. The data is biased. Yeah.
So there could be more.
Yeah, I think you're totally right. You know, we sequence less than 1% of positive cases, and I think even our positive cases, I think we have a lot of people that get COVID that don't report that they have COVID. They don't get tested. So yeah, I think this is just a very imperfect snapshot of what's going on, and I think we should sequence more so that we get a better idea of which variants exist where and how prevalent they are. Yeah, that's a great point. I think also in terms of like, you'll see that there's much more wilder swings as we get closer to today and on these plots, and that's just because these sequences have not been submitted yet. This data closer to now is a lot more sparse. So we do have to, whenever we interpret this COVID sequencing data, we have to understand that it is imperfect and that it is biased. I think also, you know, who gets sequenced and who doesn't is also a bias. There could be bias in, you know, that this is not a uniform sample of the population. So yeah, it's a great point.
Thank you.
So we have some more questions. So I'll take like 3 more of these and then I'll move on to the rest, and then I'll come back around at the end of the talk to these. That's okay. Yeah. So net, how many variants have been identified? Are there areas of the virus structure that mutate more frequently than others? Yeah, it's a great question. I'm not sure the exact number of variants that have been identified. I know it's in the hundreds. I think most variants are, you know, if we go to, skip to this next strain picture. There's different ways of defining a variant. I think on the next strain site, they actually have a wider definition of variant where there's only about like 10 or 12. I'm not sure, but much less than the notation I was using. You know, the B.1.1.7 is, I guess, like a more detailed way of grouping these. The grouping is arbitrary. Or not arbitrary, but the grouping is sort of like human defined. It's not like an intrinsic fact of nature. It's how we think about the data. It's how we group the data ourselves. And are there areas of virus structure that mutate more frequently than others? Yes, definitely. I think just like any other organism, there's parts of the virus that are amenable to mutation and parts that are not. If you think, if we think about humans as an analogy, we have a lot of genetic variation in height, because whether or not someone's taller or shorter doesn't really affect their survival, at least in today's society. Whereas we also have some areas of our body which really can't be mutated, right? Like we can't make our heart drastically bigger or smaller because that would present some really bad, medical effects. So same thing with the virus. There's some parts of the virus that are really critical for it to function that when it's mutated, the entire thing breaks down. And there's some parts of the virus that are actually advantageous to mutate, especially if it's trying to escape from vaccines or therapeutics. The spike gene that I was talking about earlier is, one of the most mutated portions of the coronavirus genome. So yeah, feel free to speak up and let me know if I didn't answer your question the way you wanted it to. I could always go into more detail. A Yang asked, I'm curious if there are certain markers or gene sequences that suggest vaccine efficacy or inefficacy if mutated. Yeah, there's certain portions of this spike gene which people there's some great work by Jesse Bloom's lab where they actually test a lot of these sites on the genome and see if they are more, I guess, not sure how to put this, but if they are more likely to affect, resistance or escape from vaccines or therapeutics than other sites. So I think they also have a really great website about visualizing this. I kind of regret I don't have a link to it in this presentation. But yeah, I think a lot of this, they actually generate that data via tests. They tested this all in the lab, and we really can't know without testing. So I think with some of this data that people generated, we can make predictions, but we would have to test it, yeah, physically. Okay, I'm going to continue, but I'll come back around to some of these other questions, just in the interest of keeping the pace going. But these are all great questions. Thank you. Okay, so yeah, for the next part of the talk, I'll just go into how we develop the site, some challenges we encountered along the way. I just want to preface this by saying that the site is very much still in development. I don't want to present our site as a success story. I think we have much to add and much to improve. I just hope to tell a story that I hope you'll find some parts of it interesting or maybe relatable. So this project started with a couple of scientists in our lab. We started this project because analyzing these SARS-CoV-2 genomes just takes way too long. It's just too hard, or not too hard, but very time consuming. So first, these genomes and metadata have to be downloaded separately as separate files from this GISAID database, which I mentioned earlier. Then we have to run this quality control. We have to filter out low quality genomes. Then we have to dig through these genomes and extract mutations. And this is actually a really computationally intensive process. After all of that, they could finally run the analyses that they wanted to run in the first place. And on top of this, the data is changing every day. These new sequences are being added every day. So whenever they wanted to update to the newest data, they had to repeat this entire process. So our goal after experiencing this firsthand was not just to streamline this process, but to, and not just to do it for ourselves, but to make this process data available to other researchers and other scientists so they wouldn't have to go through the same struggles that we did. I had to make some key decisions as I started to build the site. So first I decided to not use any of these, I guess what I'm terming like unified solutions like rShiny or Plotly Dash or Bokeh Server. I've used some of these in the past. I think while they're really great, really quick to put together, I often found I had to hack my way around problems. I found that they didn't deal with like really large amounts of data very well, which is sort of the data that we have. So instead I started with this boilerplate kit called React Slingshot, which sort of made all these architecture decisions for me. I just really went for React because I've used it extensively on some prior projects, and I've also worked with some of the other tools listed here. These might be familiar to some of you. So for plotting, we had like a ton of options to choose from. There's many JavaScript plotting libraries. I had Vega on my radar for a while, and I decided to use it because I think it's more expressive than some of these higher level libraries like Plotly, and I think not as hard to learn as these lower level libraries like D3. And another thing I loved was that Vega, we wanted the site to be interactive. We thought that Vega was really built from the ground up to be interactive, just like D3 is as well. One of the example plots from Vega that caught my attention was that someone actually wrote Pac-Man. And the way that Vega is designed, it's you define a plot with a JSON file. So someone wrote Pac-Man in JSON, which I thought was just really neat and sort of kind of proved to me the expressive and interactive power of Vega. And so next, just talking about some of the data processing. So this is the GISAID database, just a preview of the site. When we first started, I would have to actually process all of this data manually. I know that I mentioned our goal was to streamline the process, but we figured that first just putting in the manual legwork would allow other research groups to save time from doing the same thing. So well, I would download these. I would download these sequences and metadata files, store them locally in my computer. There's no public API for the site, so you just have to do this manually. And then we would manually run extraction scripts from these genomes and aggregate them all into one file. And extracting mutations from genomes is actually very, very computationally intensive, at least the way we were doing it. So long story short, it requires like mountains and mountains of RAM. And to do this, we actually had to use these high memory VMs from Google Cloud that had like 600 gigs of RAM available. And even then, some of the data processing would take hours. And then we kind of ran into the problem of like, we have these 50,000 genomes. It's a lot of data. How do we actually serve it to clients? Genomic data is quite large, so we can't just rely on some like classic solutions meant for smaller amounts of data. So we first considered this conventional database server client model. We just decided it was pretty hard. It would require a lot of work. The second solution that we considered was, what if we could just stuff all of our data into a JSON file and then just gzip it, just compress it, and then host it on like a static file hosting solution like Google Cloud Storage, which is the equivalent of Amazon S3. This is really simple, but we'd also have to engineer some asynchronous requests into our JavaScript application. I know it's not that much work, but I guess like our third solution was just to do this manually. It was just to take our data and just directly inline it into our JavaScript bundle. So package everything together into one request. And we ended up going with the last solution because our mindset, at least when we were developing this project, was that this was in May of 2020, that COVID was still really serious, that we wanted to develop this as an emergency response. Like we want to get the site out there as quick as possible so that it can be used by scientists as quick as possible. So yeah, some of these solutions were a bit dirty and hacky at first. So yeah, we did this all manually. We manually downloaded data, manually processed it, manually inlined it into JavaScript bundles, and manually uploaded each new build every day to VM on Google Cloud. I just want to quickly go over that. Like, I'm not a developer by training. I don't really know the latest trends in web technology. I don't know best practices that are employed by big development companies. So I enlisted the help of my friend Kevin, who's a senior web developer at a big tech company. He just suggested that we replace some components in our initial boilerplate app and really put in a lot of work to glue everything together. So just want to do this as a shout out that his changes were really essential to the app's development. And I'm not really sure it would have worked out without him. So yeah, after all this, we finally landed on our first functional build of COVIDCG. This is much simpler than the version of the site that's up now. It's just one plot and one table. But I think some scientists in our group found it immediately useful for their research. Yeah, so next I just want to quickly go over some additional automation that we did. So there's this really great data workflow tool called SnakeMake, which is just like a makefile, just like people use for C and C++, but for Python. It builds this really cool like dependency graph for our data, which means that if we add like a new file, it'll only run jobs for the file that changed. It'll only update the necessary nodes. So this allowed us to sort of automate what used to be like manual script running. And this drastically cut down the amount of manual work we had to do each day, although we still had to manually download data from the GISAID website. And yeah, after some additional automation, we were eventually able to make this 100% hands-off. So first of all, we were able to get access to an API from GISAID so that we no longer had to manually download data every day. We expanded our pipeline to make calls to the API and also automatically upload data to Google Cloud Storage. And we did this all once per day via a cron job. And we also moved our server from sort of manually managed VMs to Google Cloud Run, which runs off of Docker containers. So this is like an auto scaling thing that can adapt or scale to large amount of requests. Say that if our site gets published in CNN, which it did, and we got a ton of traffic, and this all just handled that fine. Whereas I think the VM would have just crashed. And so yeah, now I think we have this process mostly automated, which is great. Another really quick thing I want to go over is that we solicited some early feedback from a lot of different scientists, both from abroad and from around the world, actually. So yeah, we had many users who are currently performing COVID research back in 2020. We had them use our tool and give us feedback, and a lot of this feedback was really crucial for the vision of our site moving forwards. So yeah, after months of work, we finally launched our site to the public. We published a paper, we got some press attention, and over the last few months, we've just been gradually fixing bugs, improving performance, and have some plans to add some new features to the site. I just want to quickly talk about this data disaster. So about a month and a half ago, I was notified that our app had stopped loading, which was like a big nightmare. So I sort of jumped on and tried to see what was happening. So just for context, when we started the app, there was only 50,000 coronavirus genomes publicly released. This is about 30 megabytes the way we package the data. If we compress it, it goes down to like 2 megabytes, which is like, 5 or 10 seconds of a YouTube video. So it's a relatively small file size. And this is a big reason for why we decided to go with this like inline data solution earlier, is because we really didn't have that much data. And in May or June of last year, we figured at the current rate of submission that in about a year, we'll have maybe like 200,000 sequences. It's a lot of data, but it's still, again, it's not too much. At the end, you can see here that in real life, the actual rate of sequencing has really skyrocketed. At the time of the crash, our site was handling data from the site was sending over 700,000 genomes to our clients. So compressed, this was about like 30 megabytes. This is actually like a really big download, but for a fast internet connection, it didn't take more than a minute. Uncompressed, this data exploded to about 500 megabytes. And just as a surprising little like tidbit that I found, when you open Chrome Task Manager, I found that even after loading 700,000 genomes into the browser, our app was still taking less RAM than Gmail was, which is kind of weird. I don't know whether it says more about Gmail or us. But yeah, anyway, after looking up our errors, I found that Google Chrome refuses to parse any JSON that's larger than 512 megabytes. And at first I was actually like super annoyed at this. I was like, this is such a stupid, arbitrary rule of Google Chrome. And then I was thinking about it more. I was like, actually, this is pretty reasonable to not parse JSON over 512 megabytes. This is actually an okay limit. So yeah, now that we have like 1 million genomes, how do we serve 1 million genomes now that we can't just inline it all into a JavaScript bundle or serve just one big JSON file? It's funny because we started at sort of the laziest solution and slowly moved back to sort of the more principled solution of having a database and a server and a client, which can serve only small chunks of the data that the user requests. And this is what is live now. So this is like sort of the version 2 of COVIDCG. I was actually working on this a while. I anticipated that this would happen one day. I just wasn't anticipating it would happen so soon. So now we have data being loaded into Postgres that's communicating with the Flask server, and that Flask server is communicating with the front-end app right now. Yeah, so just lessons learned, I think. I don't know, I always think about that programmer meme where it's like, oh, I can spend 5 minutes doing it manually, or I could spend like 4 hours automating it. And I think, the answer from that meme is that like, yeah, just do it manually. Like all programmers do is like they want to automate stuff. I think that if I sat down and sort of wrote out how much time I would be spending each day doing manual work, that I would, come to the conclusion that I should actually automate much, much earlier. So I think sort of thinking about the future and thinking about what automation can do for us would have helped me a lot back then. So I think also asking for help was really helpful for me. I think I realized that I'm not really, I'm an amateur programmer. I didn't really learn this in school. It was really, I think a lot of the technical direction of the site was driven by my Kevin, the senior web developer, and a lot of like the scientific direction and the design direction was driven from these scientists that we interviewed early on in the process. And then lastly, just about scalability, that we have wasted, a lot of time just putting out fires and having to deal with these large bumps in traffic, and just having a scalable solution from day one would have saved us a lot of time as well. So I see we have questions. I'm almost done with the talk, so I'll get to them. I'll get to them soon. I know I'm running very much over time, so but I'm almost done. Yeah, for the last part of the talk, I just want to discuss the future of not just data science in the COVID field, but also all across biology, and how skilled data scientists that don't necessarily have biology expertise can help advance science and global health. So I think biology and data are growing ever and ever and ever closer. Here I'm showing the cost of DNA sequencing in blue from just the year 2000 to now, and it's outpacing by a massive pace Moore's law for semiconductors. And this is just an example of genomic sequencing. I think in every field of biology that we massive reams and reams of data being generated now. And it's not just like the 1 million SARS-CoV-2 genomes, it's also initiatives like the UK Biobank, which has sequenced over 100,000 human genomes. It's like these protein sequence databases, which are behind Google DeepMind's AlphaFold project, which some of you may have heard of. And I think that this, this massive amount of data allows people working in data science and biology to solve really interesting data problems, not just solving cool biology problems. Because I always kind of found it ironic that at least a lot of my friends are computer scientists, and they spend all their time in school learning about data structures and algorithms, algorithmic complexity, all this fundamental computer science stuff. But in their actual jobs, they never get to use this knowledge. And in biology, all these scientists who are amateur programmers actually have to put these concepts into practice because they have so much data and because they need to be efficient. And this is just like my anecdotal experience. I know I might be biased here, but I think that there's tons of really interesting data science and computer science algorithms, problems like optimizing algorithms, improving data storage, improving query performance, and even more that are not really tackled in a lot of conventional data science industry jobs. Again, I don't have that much personal experience, so I might be biased, but this is just my opinion. And I think it's not just like really technical data visualization problems. It's data processing problems. There's also really some interesting visualization problems. I think biologists aren't just amateur programmers; they're also really amateur visualizationists as well. This is an example of the really widely used UCSC genome browser, which is like an amazing tool, but it has like decidedly old school look, and it's not super intuitive to use. And then here's an example of Nomad, a tool that sort of it doesn't have the same exact goals as the genome browser. It serves a little bit of a different purpose, but I think Nomad is one of the new tools in this new age that's sort of leading the field and finding really great ways to explore complicated data like human genomic data. So my point here is that like contributing to this new age of biology and data science isn't just a purely technical thing, but a lot of these problems are actually visual. In my opinion, it requires the talents of a lot of creatives like graphic designers and visualization specialists to actually get through this piles and piles of data. And I'm just listing a bunch of, if you have specialties in any of these fields, I think that there's a really large need for this expertise in biology. Sorry, my lights just shut off. Okay, just want to thank everyone. We've had help from a lot of people, and I will take some questions now. Okay. So from Adam, what types of issues would be good for others to help with? So yeah, like I mentioned, this COVIDCG project is open source on GitHub. I also use it just to track issues and what's going on with the app. Yeah, I'm not sure that that's something on me. I think I should start marking issues as good for first-time contributors. But yeah, I think I hope that I've made the development. The COVIDCG is based on a bunch of Docker containers, and it's preloaded with some example data. So hopefully, if you want to get it up and running, hopefully that process is easy. That's something that I've tried to make easy. Do we have location data of who is getting sequenced? Sometimes. So I think depending on which country and location we have sequencing data for, the level of detail as it pertains to location and patients really varies just because of patient privacy laws that vary between different places. So I think really notably in the UK, they're extremely vague about their patients, whereas sometimes in the US, they can be a bit more detailed. But a lot of this metadata is sometimes pretty vague. I think in the UK, you won't even, they'll just say if it's from England or Wales or Scotland. They won't go any more detailed than that. Or sometimes they will, but mostly they don't.
One of the to tack onto that question, one of the things that I was curious about is the urban and rural outbreaks are often different in many ways, especially in the response. And I wasn't sure if it was like, I don't actually know when sequencing how far samples will travel before they get sequenced. So I wasn't sure if it was like things that were near Broad Institute happen to get sequenced more than things that are not near Broad Institute or something like that.
I'm not sure the answer. I'm not sure I know the answer to that question. I'm not really familiar with the logistics of of how samples go from testing centers to sequencing centers.
Yeah.
I'm not familiar with that. Sorry.
You said there's some data available. Is it data of where it was sequenced, or data of where the patient was?
Yeah, it's data from I guess where the patient is. I guess that's a complicated question because they could live somewhere and traveling somewhere else. Yeah, so actually it was really interesting because some of these sequences do have travel data associated with them. So they'll say that this patient traveled recently from this place, and that's how they actually can do a lot of this evolutionary tracing, but not with every sequence. So yeah, it's usually about where the patient lives. And sometimes it's at the town level, sometimes it's at the county level, sometimes it's state level, sometimes it's only at the country level. It really varies. It's not consistent.
Very cool. And that data is not currently visible in your tool, but is in GISAID, or is it? And I'm not sure where to click.
Oh yeah, sorry. So GISAID is the database which hosts individual sequences. Our site is serving aggregate data, so we will not display the metadata for any individual sequence just based on our terms of service.
Right.
But yeah, if you go on the GISAID site and register, you can look at individual sequences and see, any metadata about the patient, if there is any, and where exactly it came from. But on our site, we just sort of allow people to select broad locations, and then we collect all the sequences that are associated to that location. If that makes sense.
Yep. When you select a location, it's the location of the sample, not where it came from, though, because that's not or obfuscated by the abstraction. Sorry, stop answering my questions. Go to somebody else's question.
Oh no worries. Yeah, we don't always have travel data, so we haven't really prioritized processing it, but we could in the future.
Thank you for the talk. It's really nice, a really nice piece of work, and hats off to Kevin as well. I wanted to ask you to go back to one slide, which was the first instance of the UI. It had a big stream graph on it, and you were talking about how your users were really finding that valuable. This one, yeah. Can you just talk about what your users were looking for and what helped them in this view?
Yeah, so this was about, I think when we were interviewing people, it was about July or August of 2020. I'd say that we didn't really have a lot of these scary variants popping up. It was just a few mutations here and there. And we weren't actually looking, or we were sometimes looking at variants, but we were actually looking at individual mutations, which our site also allows you to do, is to not look at these evolutionary related groups of sequences, but actually drill down into the effects of, what is the effect if this specific position is mutated? Is that increasing or decreasing in frequency in these locations? So I think that's like one big part they found useful. And I think also what they found broadly useful was just the ability to sort of use our interface to filter the data that they wanted to look at, and then to download it for their own purposes. Because I think our site's not always going to provide the exact visualization they want. I think it's a really quick way to understand what's happening and then dig deeper from there. So
in a nutshell, to sort of be so naive, is they were using us to isolate what portions of the virus in the set that they had appeared to have mutated.
I guess you could approach sort of exploring this virus data from two angles. You either have a mutation in your head that you want to explore, like you have this, you might hear that like this E484K mutation in the South African variant and in the Brazil variant that you open the site and you want to see, like, is this in Massachusetts? Is this growing in Massachusetts? That's sort of like mutation-centric approach. And then you might also have this global surveillance-based approach, which is just like, what's circulating in Texas? Like, you might just want to ask that question. So I guess it depends on what your goal is. Does that answer your question?
Sure. And so what's at stake for them is the design of some sort of not an intervention, but just they're looking at this to figure out what their policy response should be.
Yeah, and I just
yeah.
Or sorry to interrupt. Go on.
I mean, what do they do once they've identified that this particular variant seems to be pretty active in Texas? What do they do with that?
So yeah, I think we would caution against anyone sort of looking at the plots in our site and making a surefire conclusion about the ground truth of things. Like sorry, I forget the name. Like one of our participants mentioned earlier that the data is really biased. It's not, it may not be uniformly sampled. It's sparse. it does not necessarily represent the ground truth. So we see this tool as sort of really rapid hypothesis checking and motivating people to dig deeper into the individual sequences. So again, like we provide aggregate data, we provide these plots that make it really, we hope, easy to sort of zip around the data and ask questions quickly. But I think for a principled analysis that you have to dig deeper.
This is about being able to focus attention efficiently.
Yeah, exactly.
Okay, good. Thank you. Thank you. Great talk. Thank you.
Sorry, just going through more of these questions. So A Yang says, did you consider any parallel pandas libraries like Dask, Modin, CUDF, and Spark? Yeah, actually, again, like I'm kind of a noob at this stuff. Like I read some forums about some of the new stuff coming out, but I don't necessarily have the time to try all of them. Yeah, definitely. I think that the snake make program I mentioned earlier. Let's see if I can find it. Sorry, I thought I had it somewhere here. Here it is. Yeah, so I mean, this I think SnakeMake is also, I guess it's also like a parallel. It has parallel processing features. We usually run it in a parallel mode to process all of our data. It's compatible with different computing clusters like Slurm or Singularity. It's compatible with those are all I know of, but yeah, there is some parallel computing power built into that. So yeah, I definitely don't run any of this on like a single thread. That would take forever. Okay, yeah, last question from Kirsten, sorry for pronounced that wrong. Any advice for someone with a biomedical background, research and clinical, who's new to the data science field? Yeah, I'm not sure if I'm the best person to answer this. Like I've been sort of programming as a hobby from high school, but I think that yeah, I guess I'm not. I think there's tons of opportunity. I think like as with the sort of the best ways to get started with programming is to get started in a project. I think that if you already have your foot in the door in a company that you have access to data that but yeah, I think that these groups like this PyData group, any I don't know if there's any rivalries between the R groups, but I think a lot of the R user groups are really great. There's a few resources off the top of my head which are great introductions to sort of analyzing data, not necessarily approaching computer science from the computer science mindset, but approaching computer science and data science from sort of the scientist perspective. Like the first thing you want to do is really plot data and not learn about linked lists or something. So yeah, I think there's, if you I'm trying to look up, there's a couple of these O'Reilly books, like Python for Data Science, I think is a really good one, where they sort of just start you out about plotting data, sort of start out doing interesting stuff that isn't super dry. I think that's a good way to start, but that's just my opinion. Okay, I'm curious if this tool can be used to figure out, or sorry, Morale asked, I'm curious if this tool can be used to figure out what exactly is driving increased cases in a particular location. Yeah, that's a great question. I'd say no, this tool cannot be used for that. So again, like what I was saying earlier is that this tool is for really like rapid hypothesis generation, maybe just like really quickly getting a lay of the land of how things look like. Even with locations like Brazil, where they have this P.1 variant coming out, and they also have this really dramatic rise in cases, this research coming out of Brazil isn't necessarily like, oh, these two things are correlated, so therefore they must be causal as well. I think everyone's really careful about that. There could be many factors that drive increase in the pandemic in a given location. It could be social behavior. It could be genetic, right? It could be these variants. It could be government policy. It could be a wide variety of things. So we can't really make these conclusions from the data on our side, at least. Yeah, Adam.
Sorry, I was like, why are you talking to me so much? I have like two super technical questions. I don't know if this is a question maybe for Kevin potentially too, but As I was hunting through, I am trying to look at all the example analysis, and one of them loads a blank page, lineages in Iceland. And I noticed that because this is React, not data, but when I go through different places, there's no history. It's all dynamically generated, so I can't hit back from wherever I am. Do you know what part of the design would need to change in order to be able to hit back and go to the page you're on before?
Yeah, that's a good question. Yeah, we should implement better routing. I think a lot of this first site was really just sort of hacked together because we need to get it out. But yeah, there's definitely many points of improvement. I welcome anyone using the site that finds any place where they get stuck or any problem, just open an issue and we'll
the problem there, sorry, Albert, the problem there is that we're using MobX, and we're using a MobX router, which is a not very well supported router. And so in order to do this, it's going to just take a little bit more legwork than if you were just to do it with standard React router, I believe. Yeah.
I will, yeah. I don't know anything about React routers. I just know I've seen React apps with history, and I think I can go Google it and try to figure out a little bit more information. I'm probably not going to be able to actually submit a PR, but if you mark one of your issues as good first issue, I'll hunt for it.
Yeah, we'll definitely have to go through some of our issues and do that.
I have one more question about genome sequencing. Not something I'm familiar with, and just the expression of the base pairs looks pretty quote unquote unintelligible. But that's a lot of memory to be using to be sequencing. And I'm curious about what makes that so memory intensive. And can you point me to one thing that might be good to read about to sort of understand what the computation of genome sequencing is about? Yeah,
I had one slide here that I cut. I'm trying to find it. Yeah, I was going to sort of explain a little bit about sort of the memory requirements of this. Before I go into that, there is a more efficient way of doing so than we are doing it. It's called NextClade. It's also from this NextStrain project team, and they wrote some bespoke code in C and C++ to align these viral genomes. And sort of like I said, it's there's a lot of inefficiencies in the way biologists write code that I think can be improved by serious programmers and data scientists. So yeah, I think the question is we have these genomes that are 30,000 characters, about 30,000 characters long. The individual characters can change. They can also be deleted, or they can also have stuff inserted. This has happened in the coronavirus genome. So just as an example here, I have sort of a reference sequence. Let's say that this is not the exact sequence, but let's say this is the reference virus from Wuhan from last year. And then we have all these different lineages where, some of the letters change. There can be some deletions. There can be, these would be technically insertions. And I think the big problem here is that we have these strings that are 30,000 characters long, and we have to find an optimal global alignment. So that we have to, imagine we first have this big sliding window where we slide 30,000 times, except then we also have this problem of insertions. So we can have certain portions, skipping others. This is what sort of causes this to be so memory intensive, because we have to solve this entire problem at once. I think the actual algorithms sort of do a divide and conquer strategy. The reason this NextStrain or NextClade, product is so much faster and less memory intensive that it can even run on the browser is that they've made some assumptions about this viral genome that made some optimizations. I'm not exactly sure how it works, so I won't comment on that, but they've improved on a more general tool. So yeah. That's sort of why this process is so computationally intensive.
Got it. Thank you.
Yeah. All right. Do we have any other questions? And check the chat. Sorry. Yeah, the GCP memory is usually cheaper than engineering time. That's funny because yeah, that's what we thought at first, is that we could just use these really beefy VMs in the cloud and just sort of brute force our way through the data. These VMs are actually quite expensive. We could spend I don't know, maybe $50 a night processing some data, which doesn't sound like too much, but it really adds up. So we actually used this problem to motivate buying just a really big workstation computer in our office. that you know, also has some graphics cards for machine learning stuff. But you know, this having a physical computer instead of relying on cloud computing actually paid for itself within like a month or a month and a half. So like the cloud is convenient, I think, more than it is cheap. Yeah.
I hope, I know Google and GCP and AWS have been throwing out credits to a lot of groups like CoronaY and other things. So Broad gets a pretty hefty discount on GCP is my guess, but I hope that it doesn't, that money is not preventing like certain things, because I feel like GCP will give that to somebody if you ask the right way.
Yeah, we haven't asked. I'm not sure if we get discounts. I know that we don't pay overhead for cloud computing, but that's it. Yeah.
So yeah, I think that's it. Albert, thanks for a great talk, and thanks for answering the questions. You know, it's just nice to be able to learn exactly how all this is done. So thanks again, and a big round of applause, I'm sure, from everybody.